The broadcast is now starting. All attendees are in listen-only mode.
Welcome everyone to the IIVS-sponsored webinar on in vitro phototoxicity assays. This webinar is provided as part of IIVS's educational webinar series and is funded through contributions to the Institute. Today's webinar will cover practical experiences and current perspectives on the use of the in vitro 3T3 infrared uptake phototoxicity assay, which is OECD Test Guideline 432, and the use of reconstructed human epidermis models for phototoxicity testing. We will provide a general overview of the use of the assay, and we'll discuss some recent information and publications. We will close the webinar with approximately 15 minutes of question and answer time. Before we start, let's take a moment to ensure that everyone is ready and familiar with the webinar control panel. Due to the large number of attendees, all audio other than the panelists has been muted and will remain so for the duration of the webinar. You may ask questions by using the question feature at the bottom of the control panel to the right. Click on the arrow to maximize the control panel and view the questions window. Technical questions can be submitted anytime, but will be held until the end of the presentation. We are recording this webinar and the question and answer session at the end. Any questions that are not able to be answered within the scheduled webinar time will receive a written response that will be distributed to all webinar participants, along with a PDF of the presented slides and a link to view the webinar recording within about a week. Let's meet our speakers. Today's presenter from IIVS is Ms. Allison Hilber. She is the study director for phototoxicity assays and has been working with the assay since she joined IIVS about 8 years ago. Allison has presented the assay many times during IIVS hands-on training workshops, both in the U.S. and internationally. We are very pleased to have Dr. Manfred Liebsch with us today as our guest speaker. Dr. Liebsch received his PhD in biology from the University of Hamburg and took a position as the head of the pharmacodynamics unit at the Laboratory of Pharmacology and Toxicology, which is a CRO. From there, he moved to ZBET, the Alternative Methods to Animal Experiments at the Federal Institute for Risk Assessment, where he rose to the position of scientific director and head of unit in November of last year. Manfred retired from ZBET, so we're very grateful to him for his participation today. I will now pass control to Allison, who will begin with an overview of in vitro phototoxicity.
Thank you, Mandy. Good morning, everyone, and welcome to our webinar on phototoxicity. We're going to start our webinar with an overview of the 3T3 Nutri-Red uptake phototoxicity and the three-dimensional reconstructed human epidermis in vitro assays. We'll start the webinar with a brief outline of these assays, highlighting their application and use, procedures, and data analyses. I'm not going to spend a whole lot of time discussing specific details, as these assays have been around for a number of years, and many of you may already be familiar with them. However, we will have a Q&A session at the end of the webinar to address specific questions you may have. Today we're going to spend some time talking about practical experiences using these assays, specifically solubility and tier testing approaches. These are some of the common discussion points for those who are working with these assays or have used them to assess photo safety. And finally, Dr. Manfred Liebsch, our guest speaker, will address current perspectives on photo safety testing, including those from a pharmaceutical perspective, UVB absorbers, and highlight some of the discussion points from the 2010 ECBAM 3T3 phototoxicity workshop. Manfred has spent a number of years working on these assays from their development to validation, and his efforts have propelled these assays to the levels of understanding and acceptance they have reached today. We're very honored to have Dr. Liebsch's participation with our webinar today. Thinking about photo safety testing, a reasonable start is in vitro assays. Why are they performed? How do they work? What assays do we have there available? Before you evaluate your test material in these assays, you want to take some initial steps to understand your material. Does it absorb in the wavelength of 290 to 700? Will it accumulate or distribute to sunlight-exposed areas on the skin and eyes? Will it produce a more reactive species? If you answered yes to any of these questions, then you've likely looked into photosafety testing. There is common ground among considerations for testing. However, each regulatory body may have differing approaches or recommendations. So it's really important to understand those requirements as you move forward. Cosmetics and pharmaceuticals, for example, are very different types of products and may require different approaches and considerations. At IIVS, we use two assays to determine phototoxic potential: the 3T3 Nutrared uptake assay and the three-dimensional reconstructed human epidermis. These assays share similar approaches to assess phototoxic potential by making comparisons between cells or tissues exposed in the presence or absence of UVA and visible light. These assays can be used independently or as part of a tiered testing approach. As we move forward, we'll individually break down these assays a bit further, and then we'll uncover how these assays might be used together as a tiered testing approach. Again, with these photosafety assays, the idea is essentially the same. We want to compare the cells or tissues in the presence and absence of UVA and visible light. There are a number of considerations in how and when to use these assays. So let's take a look at some of these general comparisons. The 3T3 assay is a 96-well-based monolayer assay utilizing the 3T3 mouse fibroblasts, whereas the three-dimensional assay is a reconstructed human epidermis tissue model with a number of cell layers, including a functional stratum corneum. Now, this is important because the function stratum corneum mimics the human epidermis with a barrier function. As you can see from the photos, the micrographs of the 3T3 confluent monolayer and the 96-well plate to the left, and then also the histological sectioning of the layers of the three-dimensional tissue provided by MatTech to the right. Again, notice that top layer with the function stratum corneum. In terms of applications, the 3T3 assay is a bit more limited with the types of materials we can assess. Typically, ingredients, botanicals, and antibiotics are good candidates for this assay system. We'll treat the cells with at least 8 concentrations of the test material up to the highest concentration of 1,000 micrograms per mil. The three-dimensional assay provides a bit more versatility in the types of test materials we can evaluate. Like the 3T3 assay, we can look into ingredients, but this assay also offers us the freedom to test materials that are insoluble and also final formulations. As our standard practice at IIVS, test materials are topically applied, although systemic approaches where test material is treated into the culture medium has also been evaluated. Viability will be assessed using the Nutrared uptake endpoint for the 3T3 and the MTT endpoint for the three-dimensional model. The 3T3 Nutrared uptake assay went through a formal validation process in the late 90s with 30 test chemicals. Its demonstrated predictivity and usefulness propelled it to an OECD test guideline in 2004. The OECD test guideline makes this assay quite appealing for use, especially when a regulatory accepted assay is preferred. The three-dimensional assay, although it went through an initial pre-validation in the late 90s, showed great promise, but a formal validation has not yet been conducted. So we have these two assays for photo safety assessment. How do we know where to start? So there's different approaches that we may need to consider in terms of hazard assessment and risk assessment. The 3T3 assay is generally recommended as a first-tier approach, depending on the regulatory agency. A negative result in the 3T3 assay may not require additional photo safety testing, and again, this depends on the regulatory agency. In cases where additional information is needed, for example, if we were to get a positive 3T3 result, or perhaps we're looking from a hazard assessment perspective of an ingredient moving into a risk assessment of a final formulation, the three-dimensional assay could serve as a second tier. I'll focus a bit more on these tier testing approaches when we move into our practical experiences. I wanted to give you a little bit of background on how we actually use the Nutrared uptake endpoint. This is what's used to assess viability for the 3T3 assay. So the cartoon on your right highlights the mechanisms involved here. NutriRed is an uncharged dye and passively crosses the cell and lysosomal membranes. The lysosomes with an acidic pH transfer charge to the NutriRed dye. Once the NutriRed becomes charged, it becomes trapped in the lysosomes. Cells which are viable and have intact membranes will be able to retain the NutriRed dye, and those cells which are dead or have membrane integrity issues will not be able to retain the dye. You can see from the microphotographs below that the 3T3 is before the addition of NutriRed, and then the 3T3 is after the addition of NutriRed. Note the accumulation of the dye intracellularly within the cell lysosomes. Now let's take a look at the general overview of the 3T3 assay. Again, I'm not going to focus on the step-by-step specifics, but if you would like more information, I encourage you to ask your questions as we proceed, and then we can address them during the Q&A session at the end of the webinar. This assay starts with seeding of 96 well plates. Each test material will receive 2 plates, one for the assessment of the presence of UVA and visible light and one in the absence of UVA visible light. Each assay also includes a positive control, chlorpromazine, tested concurrently in 2 plates. After the cells have been seeded, they're grown at standard culture conditions for approximately 24 hours. This allows the cells to obtain a confluency of approximately 50%. On the day of dosing, each plate is evaluated for proper distribution, morphology, and confluency, and then labeled with the test or control material. The cells are rinsed to remove media containing phenol red, which could potentially interfere with our light exposure, and then a buffered solution is added to the plates. A serial dilution of at least 8 doses, and again, this is really going to depend on phototoxicity potential, is prepared for each test or control material. The cells are treated in 6 replicate wells with the test or control material for 1 hour, followed by a 50-minute exposure in the absence or presence of UVA visible light, for a total exposure of 5 joules per centimeter squared. The plates are then rinsed and the cells incubated for a 24-hour post-exposure expression period, where we may see enhancement of toxicity or perhaps even a repair effect of the cells. NutriRed dye is then added to the cell cultures for 3 hours, and then the cells are lysed with the NutriRed solvent. The plates are shaken and then read on a plate spectrophotometer. Now, as we move into the 3T3 data assessment, we're going to use 2 measures to assess our phototoxic potential: the PIF, or photoirritancy factor, and the mean photo effect, or MPE. Now, we have a special program provided by ZBET called Phototox 2.0 that we're able to use to assess these 2 values. In general, the PIF is going to compare the IC50 of the dark plate to the IC50 of the light plate. Now, this can be difficult to use, especially where there's a case where you have one IC50 or both IC50s that may not be obtained. So then the mean photo effect comes into play. The MPE is going to evaluate the curve responses between the light and the dark plates. And this is especially useful when we don't have an IC50 for one or both plates. At the bottom of the page, you'll see the OECD test guideline predictions in terms of our measures of phototoxic potential as being no phototoxicity, probable phototoxicity, or phototoxicity. And again, we have different parameters for the PIF and the MPE value. Digging in a little bit deeper with the PIF, here's some examples of exactly how this is going to be calculated. So we have our graphics here on the right, which would represent 2 different plates or 2 different response curves in the presence and absence of light. We have our concentration as well as our viability. And then the dose response curves, each box here represents one of the 6 replicate wells. So our IC50 of our light plate is 0.73 micrograms per mil. And then our dark plate is 15.4 micrograms per mil. If we take the IC50 of the dark plate divided by the IC50 of the light plate, we come up with a pith value of 21.1 for this particular example. Looking next into our OECD prediction, you can see that this particular test material does have phototoxic potential as the pith value is greater than 5. Now the MPE is a little bit more challenging as it's going to look at the dose-response curves of the light and the dark plates. Now previously we were looking at individual plate data, and here we're taking both plate data and putting it together on a single graphic. So this represents our UVA response curve, and this would represent our dark response curve. The area in green here represents our mean photo effect. So essentially, if you want to think about it, in theory, if you were to have 100% viability all the way across your dark plate and 0% viability across your entire light plate, your MPE value would be approximately 1. Now on the right here, you'll notice there really isn't much separation between the two curves. This is because this isn't exhibiting a phototoxic potential. The MPE for this particular example is 0.001, close to 0, and then the MPE of the graphic on your left is 0.567. Again, if we move down to our prediction model, we could see that on the left, this test article would be phototoxic, and on the right, it would be not phototoxic. The example on the left here is actually chlorpromazine, which is our positive control for the assay. Now moving away from the 3C3 monolayer and into the three-dimensional tissue model, here are some brief overview procedures. So we're going to treat the tissues for up to 24 hours. That allows for penetration of the test article into the cells. And then after that 24-hour period, we'll remove the test material. This will ensure that we don't have any potential interference with the light. And then finally, we will do our UVA or dark exposures for 60 minutes for a total of 6 joules per centimeter squared for the cells exposed in the presence of UVA. Like the 3T3 assay, we'll have that post-expression period followed by an MTT incubation. Now, MTT is going to measure the metabolic activity of the cells, and we'll have a yellow colored MTT, which is eventually going to be transferred to a purple color by viable cells. At the end of our MTT incubation period, we'll then extract an isopropanol and finally determine our optical density values using a plate spectrophotometer. In terms of our phototoxic potential assessment for this assay, we're going to, again, look at the tissues exposed in the presence and absence of light. And if we see a 30% decrease in the cells exposed in the presence of light compared to those exposed in the dark, we'll then say that that test material has phototoxic potential. So if we look at this data set here, we have a test article with a optical density value of 1.12 exposed in the presence of light and 1.3 exposed in the absence of UVA light. In terms of percent of control, we have 82%, 96%, so only about a 14-15% difference. Based on this, this would not be exhibiting a phototoxic response. On the other hand, if we look at chlorpromazine, we can see our percent of control viability. There's about a 50% difference between the tissues exposed in the presence and the absence of light. So this particular example would definitely be a phototoxic material. And here at the bottom of the page, you can see tissues exposed in the absence of light, which you see the MTT purple coloration, which may be very similar to our controls. And then over to the right here, we have tissues exposed in the presence of UVA that have a viability reduction, perhaps maybe around 40%. So in this particular example, this test article would have also exhibited a phototoxic effect. Now moving into practical experiences, we want to talk about some of the things that we routinely encounter and also discuss how we can use these assays as part of a tiered testing strategy. Solubility assessment is extremely important for a 3T3 assay and not that big of a hurdle for the three-dimensional assay since we can test insolubles. So we want to make sure that we're able to have a test article dilution that we can work with in our monolayer cultures. We're going to perform our solubility assessment prior to the assay conduct. And we have a number of different solvents that we can use. We'll start out typically with HBSS, followed by DMSO, ethanol, and acetone. There may be some cases where you have a test material that would be dissolved in something other than one of these mentioned. However, it's important that if we do proceed with that we also assess the phototoxic potential of that particular solvent. The key here is we want to avoid precipitate when possible. Heating and sonication are some of the standard measures that we do to further solubilize our test materials, and also selecting the most appropriate solvent can also help with your solubility. Now, we have seen that homogeneous suspensions can be tested, and they also do exhibit appropriate responses. The key word here is bioavailability. We have to make sure that our cells are available to interact with that test material. So you can see on the right here, we have a graphic representing our test material dissolved in DMSO first and then transferred to HBSS. You can see how the test article has precipitated out upon transfer. It's important that things like that are assessed prior to moving forward. We deal with a number of different types of materials in the 3T3 assay, and some of these responses here are things that we might typically see. We really have to step back and evaluate what we can move forward with and what we really need to determine perhaps that we can't proceed. So starting here on the left, we have a biphasic material. You can see the test material floating on top. This would definitely not be test material that you'd want to work with for the 3T3 phototox assay because you really can't be assured that if your cells are at the bottom here that they would be able to interact with your test material. Another example would be we have floating particles. How much of that is actually in solution, if any? Then we may have some test article adhering to our dilution tube. In some cases, we'll get a homogeneous suspension or solution, and typically those two types of dilutions are ones that we've found we're able to work with. Homogeneous suspensions have been shown to produce cytotoxic effects and have shown ability to determine phototoxic potential. And ideally, the best case scenario would be our solution. So what do we do when we have an insoluble test material? I like to call this the solution to the suspension. So the first step is going with our solubility assessment. Can we work with this test material? Is it homogeneous? If it's completely insoluble, like for example the biphasic material, we may decide to go all the way to the end point here to address other platforms, for example the reconstructed human epidermis. But in general, if we're able to get somewhat of a homogeneous suspension, we may proceed with the dose range finding assay and evaluate those results. If we do see some evidence of bioavailability by perhaps some level of cytotoxicity, we may then proceed with the definitive assays. However, in some cases, we have to start thinking about bioavailability. Is the test material bioavailable? If we see a viability of 100% across the board, is that absence of cytotoxicity truly an absence of cytotoxicity, or is it because the test material hasn't been able to interact with the cells? So we may determine that the test article is incompatible with the test system, and finally move into our reconstructed human epidermis model. So here are a couple examples of what data could look like in a case where solubility affected our final result. So in the example here, we have curve upswings. So you can see as we start increasing our concentrations of test material, we start to see a drop, and then finally this spike at the end of our dose response curve. That perhaps is due to residual precipitates interfering with our NutriRed, or maybe even making the optical density readings, the media, a bit cloudy. The other example here is perhaps maybe one of the worst case scenarios. So you can see after the plate's been exposed to the Nutri-Red in the cell's life, you can see the variations in the Nutri-Red uptake throughout the entire plate, and you might get data that looks something like this. So you can actually plug it into your Phototox software and get some numbers, but again, in looking at this data, this is too noisy and you would not want to proceed with this particular test material in the 3T3 assay. So let's assume that we did get those results from the previous slide. How do we move forward from the three-dimensional into the three-dimensional assay? So the important consideration here is, will the tissue tolerate the test article for up to 24 hours, followed by that 21-hour post-expression period? Again, there are multiple ways to look at the reconstructed human epidermis, but we'll stick with the idea that we're working with a single formulation here and want to move forward with perhaps the 24-hour test article exposure. So what we routinely do here at IIVS is we'll recommend a time to toxicity pre-screening. This will give us some information about the toxicity response for that particular test article. So here we have our exposure time and a % of control. So we'll look at perhaps 3 to 4 different concentrations, and based on the information, determine what would be the most appropriate exposure time to work with for the three-dimensional assay. So in this case, if we were to go with a 24-hour exposure, you can see that our mean % survival for the test article exposed in the presence of light would be about 2%, and that in the absence of light is about 18%. If we look on the prediction model of 30%, we would say that this would not exhibit a phototoxic response. However, the test material is too toxic, and we need better resolution. So if we bring it out and look at the 8-hour exposure, we have a 50% viability exposed in the presence of UVA and about 85% in the absence of UVA. So now we can correctly identify this as a phototoxic potential. So now we've seen the three-dimensional, how it could be used as an alternate. Let's think about how we can use these as a tiered testing approach. Ideally, the 3T3 assay could serve as the first tier, with a three-dimensional assay as the second tier. From the 3T3 to the 3D assay, the results could give you insight from a hazard assessment to a risk assessment. This approach could be used to model end-user experience when using final formulations, for example. Although negative results in the 3T3 assay may not require additional testing, perhaps you'd want to evaluate the ingredient in its final formulation to confirm the 3T3 results. Manfred's later going to talk a little bit about materials which are over-predicted in the 3T3 assay. So the three-dimensional assay could serve as that second tier to confirm the positive result or even rule out an over-prediction. Here's a specific example of a tiered testing approach using the 3D and 3T3 assays. This was recently published early this year, and it took a look at some common UV filters like avobenzene, octyl salicylate, and octocrylene, and also vitamin A. And we looked at both the 3T3 and the three-dimensional assays. So the compounds initially were evaluated for phototoxicity using individual chemicals in the 3T3 assay. Avobenzene was detected as phototoxic per OECD test guideline 432. And also, vitamin A did show some potential for phototoxicity in 2 of 4 independent trials. A number of those UV filters and vitamin A combinations were then assessed in the 3T3 assay. Those results showed a synergistic effect and levels of enhancement of phototoxicity depending on the combinations used, specifically those combinations that were containing avobenzene. So the next step was to evaluate those same combinations in a three-dimensional model. In the three-dimensional model, for example, we saw that the same effect wasn't seen up to 10% in one of the combinations. So when thinking about this, the combinations may have only represented a portion of those presented in the paper, but generally These combinations were 3 UV filters with or without UVA. So you can see here the 3T3 results. Here's our PIP values and our MTE. And these are varying levels of the filters with or without the UVA, or I'm sorry, with or without the vitamin A. And you can see some of these were phototoxic responses or probable phototoxic responses. Then moving in the reconstructed epidermis model, we have our concentrations up to 10% here, and our MTT as a percent of control. Which was phototoxic in the 3T3, was not phototoxic in any of the concentrations in the reconstructed human epidermis model. Another tiered approach, which was presented at SOT this year, was a collaborative effort between IIVS and Avon Products. And they wanted to, I guess, point out how they're using the in vitro results for product development. So this figure represents their testing strategy for evaluation of ingredients. So the first step is to determine if the material absorbs light. If it did, the first tier was the 3T3 phototoxicity assay. Any positive results in that assay would mean that the ingredient would no longer be used or assessed for potential use as part of a cosmetic. If it was negative in the 3T3 assay, it then went into a confirmatory clinical phototoxicity assay. If there was potential to be phototoxic in the clinical, also it would stop. However, if it was non-phototoxic in the clinical, the material was determined to be non-phototoxic. We looked at 68 different ingredients, and of those, a number were phototoxic or probable phototoxic, and many more were non-phototoxic. Here's a couple pictures of some of the botanicals that actually showed phototoxic potential. Here's a table, or I should say a portion of the table that was used. You can see here that some of the botanical blends or botanicals that were used, the 3T3 in vitro scores based on the PIP and MTE, and finally a conclusion of either not phototoxic or phototoxic. 8 of those chemicals then proceeded to the clinical setting, and the clinical setting also confirmed the results of the 3T3 assay as non-phototoxic. At this point, I'd like to pass this over to Manfred to discuss photosafety testing perspectives and discussions of the XBAM workshop held in 2010. Manfred?
Okay. Thank you, Alison, and hello to everyone, and thank you for giving me the opportunity to talk a bit about the regulatory perspectives now. For being with you, I missed the talk of your president, who is currently in Berlin, giving a talk at the Brandenburg Gate. It is his first visit of Germany since he became president, and but my wife will listen, and the internet gives me the opportunity to know what happened also this evening. Okay, so I will talk a bit about the concerns of pharmaceutical industry, and that is a bit different to cosmetic industry, where the two-tiered approach that Alison has just mentioned nicely is almost established in most companies that are using ingredients that may be phototoxic or contaminated with phototoxins due to their botanical origin. In pharmaceutical industry, each new chemical that, or each new drug candidate substance that absorbs significantly has to be tested for phototoxicity, and the concerns were that too many materials turned out to be positive in a 3T3. And we had an FPR survey in 2010. That FPR is the European Association for, of the Pharmaceutical Industry. And I will show this outcome of this survey. And as a way forward, we had in 2010 a workshop co-organized from FPR and ECVAM in Italy, and the outcome of this workshop was published, and it's now taken up first by the European regulators for pharmaceuticals, that is the EMA, European Agency for Medicinal Products. They published it as a question and answer paper in 2011 on their website, because it is now usual that we don't have special European regulations. We just get this harmonized at the ICH level, that is the International Conference for Harmonization, and they generated a new guideline called S10 on photo safety testing. This is currently in step 3 of the whole procedure. That means it has been consolidated by the ICH experts and is now in an open, or has been in an open consultation process, which is finished now, and it should be published, the final document, in 2013 in June. So that's very soon. Next slide, please. Next. Do you hear me? Thank you. Okay, so this is a bit more in detail what were the concerns of pharmaceutical industry. The first concern was that many drug substances absorb UV in visible rain, in visible light, between 290 and 700 nanometers, with an absorption which is called molar extinction coefficient of larger than 10. And of these, about 50% were then positive in the 3D3, triggering additional in vivo testing. The 3D3 was regarded obviously as too sensitive, and the approach was to desensitize the 3D3 assay by either increasing the threshold for phototoxicological testing, that means the absorption threshold, or by changing the prediction model, that means setting up the cutoffs that Alison has nicely shown, the PIF of 5 and the MPE of 0.15, and probably by reducing the maximum test concentration. So the first response of the competent authorities, the European Agency for Medicinal Products, EMA, was, your concerns may be correct. However, since negative results in the 3D3 are valid for waiving the need of any further phototoxicological testing, and that is complicated tests like photoallergy, that you only can test in vivo, photogenotoxicity, where we have totally oversensitive in vitro assays, and photocarcinogenicity, where we have several models that are very complicated and not very predictive. So the reduction of the oversensitivity should be limited to still keep a level of 100% sensitivity, because only if you have no false negatives, then you can say you are done with the 3D3 assay if you get a negative result. That is a very important point. Next. Next. Okay, thanks. The concerns of FPR were published in 2011, and I have summarized here the main points of this publication. 10 European FPR members participated in this survey. 361 drug substances were analyzed that had been tested, almost all of them in the 3D3. 97% had been tested in the 3D3. 11% had been tested also in vivo in animals, and 1%, that means 5 substances or drug substances, had been tested also in humans in clinical studies. Next, please. Thanks. So these are graphs from this publication, the basis of the concerns you see on the left side was the percentage of positive results in the 3D3. The 361 is the pie chart on the left top. The green part is the negative 3D3 assays. The red part is the positive ones, and the yellow part is the equivocal part, that is the probable phototoxicity in the range between 2 and 5 regarding the PIF, and 0.12 and 0.15 regarding the MPE. On the top right, you see the distribution of positive, respectively equivocal positive results over companies or across companies, and you see that some companies have really high percentages, like over 60%, and some companies have considerably low, like 20%, and on average it was something like 50%. On the bottom left picture, you see those chemicals that were either negative or positive, tested or equivocal, tested in vivo models. And I have to emphasize, these in vivo models are all not, let's say, fully validated. There's a lot of different in vivo models. I'm not talking about them. I've done them in the CRO 30 years ago, but my opinion is that they are not all really well developed and not validated. And that's why the OECD stopped the whole in vivo approach in 1991. What you see on the bottom is that of the positives of the pie chart, 26 were tested and only 4 were positive and the rest was negative. So, and you see the negatives, they were all turned out also negative in vivo. That means the 100% sensitivity is okay, there's no false negatives, but obviously a high percentage of false positives. My first criticism is why, if they have 50% positives, why don't they have something like shown in vivo data of 50% of 361 chemicals tested in total. So that is a bit a weakness of this study. On the right hand, you see of the positives in vivo in the 3D3, the clinical data where 5 materials were tested, all negative. Next, please. So this is also from the FPR survey, a distribution of the PIF values across all tests, all 361 tests. And you see that the distribution is quite different. We had in this graph shown one company separately because they didn't participate in this survey. And as you see in this one company, there is a quite, let's say, nice distribution from the majority being at PIFs at around 10 and a minority only with PIFs 1,000. But if you see it across the rest of the companies, it's an up and down, and therefore not really showing a certain picture where you could say, well, the PIF probably could be shifted, the prediction model could be shifted to get a bit more desensitized, or to achieve a desensitization. My critics, and that is something that I said at the workshop, that is also in the workshop report, and it is in the conclusions of the workshop report, which I would recommend to read, anyone who wants to know, is that the survey only looked for the PIF values but not at the concentrations at which these were reached, because it is, like Alison said nicely, it is very important that you check the bioavailability of your substance in the skin. And if you have a drug candidate, you will always have to do a biokinetics, pharmacokinetics, where you know finally what is the concentration of my chemical in the skin. Does it accumulate in the skin? Does it stay in the skin? And these values are absolutely necessary. And unfortunately, in the FPR survey, FPR has forgotten to ask the companies for these concentrations. So the concentration at which you get a positive PIF are much more relevant than the PIF value itself. The PIF value does not correlate with the potency of a phototoxin. That is something that we have already known in 2000 in the first ECVAM workshop, and it is if people read this workshop report, it is said there already there is no correlation between the PIF and the potency, and therefore it is not easy just to say, well, this is the distribution of the PIF. It would be nice to know at which concentrations these PIFs were obtained. Next, please. Next, yeah, okay. So, as Alison said already, absorption is the relevant trigger, and we have here the original text from the OECD guideline. Alison also mentioned that I was heavily involved in the development, validation, and also regulatory acceptance of this assay. So at that time I was in Paris, seconded as a German expert to the OECD, and I worked on 4 new alternative methods, and one of them was the 3T3 phototox assay. And in the commenting round of this guideline, commenters asked for give a specific value, because we only said a significant absorption is always an indicator for necessity to test phototoxicity. So I tried to find something, and the only thing that I found was one guidance document on photostability where we had the cutoff of 10. So I sent this around the world and asked all the experts involved in validation and involved in the commenting, are you happy if we take this value in the guideline? And they said, well, we don't have anything else, so take it. And that's the reason why it was set to 10, but it was not part of the results of the validation study. And therefore, the critics may have been correct. Next slide, please. Next. Okay. This is one of the responses of industry on the question of whether I call it now MAC, molecular extinction coefficient, whether the MAC of 10 is a good cutoff, a valid cutoff. And it's a very nice publication from Pfizer, from Henry, Foti, and Alsante. And this had 2 objectives. One, to check the molecular extinction coefficient, MAC, compared to 3T3 results, other in vivo results, and other in vitro results, and all knowledge about phototoxicity in humans. And the other was using data on photostability, that is the physicochemical stability when a material is irradiated, whether this is a good trigger. Next, please. Next. Okay. Here you see one figure from this publication from Henry et al. And these were 35 well-established phototoxins. They have been tested according to what we have in the OECD test guideline, with an absorption spectrum and the molecular extinction coefficient determined according to OECD test guideline 101. That means that you determine at each peak of your absorption spectrum the extinction in a 1 centimeter cuvette in PBS at buffered to 5.5 and 7.4. That is the range in that is expected in physiological tissues. If you can't solve it in PBS, you use acetonitrile, which has no absorption as a solvent. And what you see on this graph is, and you have several points per chemical, that except one point, all the determinations were between 1,000 and 10,000 and none in the area of 10. This one point that is below 1,000 is one peak of anthracene, which was determined at 290 nanometers. So based on this, the ECVAM workshop agreed on raising the trigger to test with a 3T3 assay or to test with any photobiological assay only materials that absorb higher than 1,000 instead of 10, which is what is still in the OECD guideline. Next, please. Next. Okay. This is just the cover and title of our workshop report, which was published in Regulatory Toxicology and Pharmacology, where FPA, organized by ECVAM, met in Italy with all the experts involved in the validation study, involved in the acceptance process. Next, please. Next. So the objectives of the ECVAM workshop were to discuss improved triggers for phototoxicological testing. One was to increase the MEC threshold. The second one was inclusion of a second trigger, and that the second trigger is the generation of reactive oxygen species, ROS, which was a new assay developed by Japan, and the Japanese colleagues had already done several interlaboratory small-scale trials and were just planning in 2010 a formal validation study, which is now finished and currently peer-reviewed. The second important point was discuss scientifically acceptable means to desensitize the 3D3 assay by either modification of the prediction model, and second, probably by reduction of the maximum test concentration. The third objective was discuss the role of other in vitro assays, in particular human 3D skin models in phototoxicity testing. So next slide, please. Next. Okay, thanks. So these are the main outcomes of the ECVAM workshop based on the work of Pfizer. That is the Henry et al. Publication that I just showed. The MEC was shifted from 10 to 1,000. Fine. Based on the data that we were shown for the photo-ROS assay, the photo-ROS assay was regarded as a promising second trigger in addition to the MEC, probably even better than the MEC, because that was very interesting. Although in theory ROS does not be generated by all mechanisms of phototoxicity that we exactly know. It is obviously with all mechanisms always some ROS is generated, and therefore the Japanese were encouraged to perform their formal validation study and to probably get this into the ICH guidance. The gray zone that Alison showed, which is currently also the OECD guideline, between a PIF of 2 and 5 and between an MPE of 1.2 and 1.5, sorry, 0.12, sorry, this is a typo. It's 0.12, of course, and 0.15. This may be obsolete, and I'm telling already now that the ICH guidance document now says you may regard this as negative. The reason is that there, in the validation study, there was a plateau of the same classification level with regard to sensitivity and specificity was reached in a plateau between 2 and 5 with a PIF between 0.12 and 0.15. And the OECD commenters, which were often regulators, they said, well, why don't you take the most sensitive thing as already probable? phototoxic. And then when you have an increase in your sensitivity above 5, and then you take it as definitely phototoxic. So this probably phototoxic was, let's say, an agreement zone, but it was not that important, and people to desensitize now take it away. The highest test concentration of 1,000 may also be too high, because what we know from another study that is not discussed today, that was also a validation study that we performed in 1998 and was published in 2000 in ATLA, is a UV filter study. From that study we know that the concentrations at 100 already are sufficient, but people requested that we analyze all the different validation phases together, which was more than 70 chemicals tested in 3 phases, and we take the cutoff from there, and that showed that 1,000 is not increasing the false positives compared to 100, so it was 1,000 was used for pharmaceuticals. It's obviously too much, but before pharmaceutical industry doesn't show these the data, regulators would not accept to go below this maximum testing concentration if it can be achieved. If you look at the ICH guidance, it says it can be lower if you show that your biokinetic data show that the concentrations reached in the tissues are much, much lower. Then they would not request, even if it is doable, these incredibly high concentrations. So in contrast to cosmetics, where the exposure is always topical, the 3D skin model may be of limited value as a second confirmatory tier test for systemic drugs. It may be, and if you read the ICH guidance as it is now, it says for dermotopical or dermopharmaceuticals, it seems of the same value as in the cosmetics area, because the kinetics are nicely modeled in the barrier model, but with a systemic application in the media, it is, let's say, a bit poor modeling of what systemically happens if a drug is entering the skin via the systemic route. EMA planned then at the ECVAM workshop to publish a question and answer document on their website, which happened in 2011. And it should be valid until the at the international level, excuse me, the ICH S10 guideline on photo safety testing is adopted. Next slide, please. Next. Okay. This is just one of many, many publications of the Japanese studies, pre-validation studies and formal validation studies on the photo ROS assay. In this study, this is the first publication of the formal validation study. In the study, 23 phototoxins and 90 non-phototoxins had been tested. No false negatives were shown. That means whenever the ROS assay said you should do a photobiological testing because it's positive, it was positive then in the follow-up testing, biological follow-up testing with the 3T3 or other assays. The number of the number of irrelevant positives, I would call it, that means positives that have a potential, but then which is doesn't turn out in vivo, that was 20 to 40% less than if we look at only the molecular extinction coefficient. So it is probably a better trigger than the MEC, and it is currently mentioned in the guidance document because the study, the validation study is finished, but it's not reviewed yet. and therefore maybe it will stay with the text like it is currently, that it is probably a better trigger or at least a second valuable trigger for phototoxicological testing. Next. Next. Thank you. So this is only the EMA question and answer paper that was regarded as an interim, so that industry would know we can we only have to test chemicals that have a MAC higher than 1000. We can stop any photobiological testing if the 3D3 is negative. All these things are put down in question and answers. And now we are in the situation that we have the ICH S10 document finished by the experts of the ICH. Next. 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 Please. Thank you. This is the ICH guidance document, as you can download it from the EMA website currently. That is on the ICH website there's the step 2 version, but we are already in step 3, as you see on the bottom. March 2013 was the end of consultation. It should be published, the final version should be published now in end of June. And from the Committee of Medicinal Products for Human Use. And this guidance document now considers all the points that were discussed at the FBR ECVAM workshop in 2010. And on the next slide, that is very busy, I've forgotten on, unfortunately, one point that is important, but I will mention it just in words. Next, please. Next. Okay, this is a very, very busy slide, and it only gives, and I wanted to show you on one slide original text examples from the current ICH S10, and it says in the first point that the MAC is now increased to 1000 from 10 to 1000. It also said that the photo-ROS assay may be a valuable trigger. It also says newly that a single-dose tissue distribution study with animals assessed at multiple time points after dosing will generally provide an adequate assessment of tissue drug levels in the potential and the potential for accumulation. These studies should be done quite early so that you can interpret your data, your in vitro data, in phototox studies much better, because you know the levels that are reached in skin, which is the only skin and eye, sorry. Skin and eye are the tissues that are exposed to light, and therefore these concentrations reached there are relevant for interpreting your in vitro data. It also says something which is important. Metabolites are generally do not warrant separate photo safety evaluations, because we don't know any case where metabolism creates a new chromophore. So that means your metabolism can change your chromophore a bit. It may modify a phototoxic potential, but it may not generate a phototoxin. That is very important, because people always say, oh, the 3D3 has no metabolic system. It is not important in phototoxicity, because that is a physical-chemical property. Then sensitivity, it says, however, to support the integrated assessment strategy described in this document, it is most important that non-clinical photosafety assays show high sensitivity. That is, produce a low frequency of false negatives. That means probably a few percent would be accepted, but a desensitization of the 3D3 assay down to 80% or something would not be accepted, because then 20% of probable phototoxins in vivo would not be tested adequately. It says also something about the 3D3, that it's the most widely used assay, and it's currently considered the most appropriate in vitro screen. And then a bit later it says in 2 paragraphs something about the skin models, which is a very nice text, and it says A, they have an important role as a second tier. B, it says if you have a material absorbing only exclusively in the UVB, you should probably directly test it on a skin model, because the skin models tolerate more UVB than the 3D3 cells. That's what we know. Of course, then you have to filter your sun simulator differently. We know a filter that we can use with the Sol 500, the lamp that we are using for the 3D3, but maybe other filters for other equipments. Next slide, please. Okay. A question that is often, and this is now a bit a change from the ICH guidance document, but to something that is almost every time asked by people, is can UVB absorbers be detected with the 3D3? And I said, if you have an exclusively UVB absorber, then you should probably directly test it on a skin model, maybe also on the photo RBC assay, that is the red blood cell assay, because the erythrocytes are also very resistant to UVB. But what people some people do not understand is if we are used a lamp that was filtered with a cutoff of 320 nanometers, that means 320 nanometers, we had a transmission of 50% still. And then it is depending on the steepness of the transmission curve of your filter, how much UVB is still present in your light. It's also dependent on the irradiance above the filter, how much UVB is present on your light. So there is a little amount of UVB, which is quite significant and good enough for stimulating UVB absorbers if they are not exclusively UVB. And what you see here is a plot of the OECD recommended reference chemicals, and you see that they have different absorption spectra, and the important, let's say for this point, important are chlorpromazine and hexachlorophene. They should be correctly detected positive in case of chlorpromazine, hexachlorophene negative, and both have similar absorption spectra. And if you look at the absorption spectra, both have their maximums in the UVB range. So it is not true that the lamp is, because we said in our publications, practically devoid of UVB. That is interpreted by some people as there's no UVB in it. No, it's not true. There is UVB, but you have to limit it very strongly, because the toxicity of the UVB light is doubling with each reduction by 10 nanometers wavelength. So if you go from 290 to 280, your toxicity of the light is doubling. And from 280 to 270, your toxicity is doubling. And that is something that is so limiting also in vivo assays, that people always have to filter great parts of the UVB away. And because it's it's phototoxic itself. Okay. So that's what I wanted to show with these as an example, that you shouldn't be afraid that UVB absorbers cannot be tested. Yes, they can. Next slide, please. Next. Okay. This is my last slide. This is actually one representative of ICVAM or NISATM from your country, Warren Casey, and I were advisors of the management in the management team of the Japanese validation study for the PhotoROS assay. And this was the second meeting that we had in last year. you see Warren Casey with the blue shirt and myself also with the blue shirt. Obviously, all the Japanese have white shirts. And so what I wanted to say is almost 10 years after adoption of the 3T3 assay at the OECD level, we still have issues to solve. However, the assay has gained a central role in the photo safety assessment strategies, and therefore all efforts are worth it. Thank you very much.
Thank you, Manfred.
All right. So thank you, Alison and Manfred. We are a little bit over time. I just wanted to hop in and let everyone know that if you type your questions into the question pane now, even if you can't stay on the call, we will make sure that Alison and Manfred address them. But we will get to a couple questions here as soon as Alison finishes up with her summary.
All right. So today we spent some time discussing in vitro assays commonly used for photo safety assessments. There's a number of important considerations, including how will your test material be used? Is it an ingredient or a final formulation? How could solubility affect your testing strategy? How will you test your material be applied? Systemically, topically? What points would favor one assay over another? How can we use tiered testing approaches? And importantly, how you can use these assays as part of your photo safety testing program. Both of these assays are individually unique, yet share similarity in their assessment of phototoxic potential by comparing cells or tissues exposed in the presence or absence of UVA light. An OECD test guideline for the 3T3 assay was adopted in 2004, and currently this is the only assay that's standardized and validated for photo safety. Manfred highlighted some perspectives discussed at the 2010 ECFAM workshop. He also talked about possible recommendations moving forward and comments on the statements recently released by the ICH. As Manfred discussed, one of the biggest concerns with this assay is its overprediction potential, especially from a pharmaceutical perspective. Yes, this assay is a sensitive assay, but it can give us a high level of confidence in our negative results. We have a number of references available. And finally, I'd like to thank everyone for your participation in today's webinar. I'd like to also thank Amanda Ulrey for her coordination of the webinar, and a special thank you to Manfred for his time, effort, and invaluable insights.
Great. Thank you, Alison. So as I said, I think we'll have time to squeeze in just a couple questions here, and then just as a reminder, the rest we will still answer. We'll just do that via email. The first question is, at what stage of drug discovery should phototoxicity be tested?
Manfred, do you have insight on that one?
Yes, it's actually stated in the ICH guidance document, but I must admit that I haven't opened it currently, and I think it was phase 2 study, before phase 2, but it is, I definitely know that it is stated in the ICH S10.
Okay. The next question is, where can I find the Phototox software for the 3T3 assay?
You can, OECD has the software available for download online. What we'll do is post a direct link in the question and answer that we send around to everybody at the end of the webinar.
Next question is, will the RHE model be validated in the near future as a confirmatory tool for the 3T3 assay?
Manfred, you might have a little bit more in terms of validation efforts for that.
Yes, the point is we have performed an ECFAM pre-validation study in 98 with Procter & Gamble and Bayersdorf. It was a blinded study, very nice data, but unfortunately at that, I must now say something very private. In this year, both of my parents died, and I was only able to publish a validation study on on skin corrosion, which then was basis of an OECD guideline, and I was not able really to publish these data. And then all of a sudden, industry just picked this assay up, and CROs picked this assay up, and since they usually use it as for in-house decisions, like, as for example, BioStuff checks all ingredients regularly with the 3D3 and the skin model if the 3D3 is positive, because whenever you have plant constituents or botanical, it may or may not be contaminated with phototoxic contaminations. Therefore, you do it batch-wise, and it's an in-house decision whether you refuse the batch or you use it because of phototoxic or non-phototoxic potential or properties. We said, well, probably we don't need an OECD guideline for that because it's used for safety decisions in-house of companies. Nevertheless, I think it would have been worth, but for me now, from a retired position, it's not very easy to stimulate a formal validation study. And also, as you may know, there's international discussions that in future we will not need any kind of formal validation because there's no validation centers anymore that really provide the knowledge, competence, and money of state-funded money for formal validation studies. They are usually done by industry now. Many of them are good, but for these type of studies, it's not so easy to achieve regulatory acceptance then.
Okay. And did you have anything to add about the software itself, the Phototox software, Manfred?
Yes, Alison said you go to the OECD website. What I wanted to recommend to anyone who wanted to use the software, download, please, not only the software but also the validation data sets. The reason is this is the only validation study that I know where all the data, except for one laboratory in the second phase, that was Unilever, because they had handwritten results that we could not easily transfer into the software. But all the validations data for all laboratories, for all runs are there as PXP files. So if you install the software in your in-house environment and then you run it, probably you run the assay under GLP, people will come, a GLP auditor will come and say, how can you show the validation in-house now? And so what you then do is you just use a data set from this validation study which is already fitted, which is downloadable there, and use the pure raw data and let them fit again in your environment. And if you get the same result as it is in the OECD downloadable files, then you're done. That's a question that I often got from CROs that set up this assay and then get into the first GLP audit. That is one point. The second point is, please download also the publication of Peters and Holzhütter of the validation of this software, because it helps you to understand why if you evaluate the same chemical 3 or 4 or 5 times, you always will get a slight different value in the third digit after the comma. It will not have an influence on your on your prediction, but it is based on the procedure that is used. That is a bootstrapping procedure. And I'm not talking about now. It will take too much time about this procedure, but it's a very important procedure, and it turns out that each calculation newly is generated by a random selection of curve pairs of irradiated and non-irradiated pairs that are generated from your inherited variation of your experiment. That is a bit mathematics behind, but it's it's really helpful to download this publication as well, read it. It is very simply written and easy to understand, and it helps you to understand the results that you get from the software.
Thank you for adding that, Manfred. That actually answered another one of the questions that we had received. So that was good information.
Mm-hmm.
At this point, I'd like to thank Allison and Manfred and all of the attendees for staying with us and your participation in the webinar today. We would like to remind you that any of these questions that you've sent us that weren't answered during the Q&A time will be addressed by the presenters, and the answers will be disseminated to all of the webinar participants within a few business days, along with a copy of the slides and a link to the recording. If you'd like additional information on the assays or help determining the best strategy for testing your compound, please contact Allison directly. Her email address is right here on this slide. And don't forget to visit our website, www.iivs.org, to find out the date and topic of our next webinar and other news in the field of in vitro toxicology. Thank you once again for your participation, and we hope to see you in a meeting, training, or during our next webinar. Thanks a lot.